the chapter Anatomy of Flowering Plants. As we have completed Morphology of Flowering Plants, today we will be dealing with this chapter, and in this chapter, as per the new NCERT, the syllabus is less, but for a safety purpose we will be talking about the old NCERT also. So let's go into the basics of anatomy. Okay, first there will be there are two kinds of tissue. One is meristematic tissue, the first one is meristematic tissue. Meristematic means which is repeatedly dividing. Then the second one is a permanent tissue, which means it's permanent. It's not dividing or replicating further. Okay. If they are dividing, it's meristematic. If they are not, then it's permanent. And now coming into the types of meristematic tissue. Types of meristematic tissue, there are mainly two types of meristematic tissue. Primary meristem and secondary meristem. Okay. And in the group of primary meristem, there is apical meristem and intercalary meristem. Clear? Okay. Now what is the definition of primary meristem? It is the meristem which is seen early in the life of a plant. Okay, which is primary. Primary means early. So which is seen early in the life of a plant is primary meristem, and it also contributes for the formation of primary plant body. Okay, so the primary plant body is formed by the primary meristem. And what are the types of primary meristem? Apical meristem and intercalary meristem. Now going into the second type of meristem, it is secondary meristem. Okay, where is this secondary meristem present? These secondary meristems occurs in the mature region. Where it present? It occurs in the mature region. Then these are responsible for producing the secondary tissues. Primary meristem was responsible for producing the primary plant body, but secondary meristem are responsible for producing the secondary tissues. Clear? And they appear later than the primary meristem. It's obvious. Okay, clear. And now what are the types of secondary meristem? The types of secondary meristem are fascicular vascular cambium, interfascicular cambium, Cork cambium. So these three are the types of secondary meristem. So be clear about it. Primary meristem, it's apical meristem and intercalary meristem. Secondary meristem, it's fascicular vascular cambium and interfascicular cambium and Cork cambium. Clear? Now going into more details of apical meristem. Okay. During the formation of primary plant body, some specific regions of apical meristem will be produced. Clear? They are dermal tissues, ground tissues and vascular tissue, which will be dealing further detailed in the chapter. Okay. So root apical meristem, as the name suggests, it is present in the root. Okay. It is present at the tip of the root. Then, okay. Shoot apical meristem, it will be present in the stem axis. Clear? Now during the formation of leaves and elongation of stem, now we have studied shoot apical meristem and root apical meristem. Now if during the formation of shoot apical meristem, there will be some cells which are left out. So the cells which are left out will form buds. Okay, the cells which are left out from this process will form buds, and those buds are known as axillary buds. Those buds will constitute to produce axillary buds. Formation of leaves, and during the formation of leaves and elongation of stem, some cells will be left behind. So the cells which are left behind are known as axillary bud, which constitute together to form axillary bud. Clear? Now the second type of primary meristem, it is intercalary meristem. Intercalary meristem. Clear? First you have to know a basic about the name, then you will get the answer. You will be clear with the concept. Okay. The meristem which occur between, inter means between, so which occur between mature tissues is known as intercalary meristem. Clear? Now this is a PYQ. This was a PYQ. The grazing part, the grazing animals will graze the grass. So the parts which are grazed by the animals are part of intercalary meristem. This was a PYQ. Okay. They occurs in grass and regenerates. These parts are the tip of the plant. Tip of the grass will be eaten by the grazing animals. So it will be regenerated. This regeneration is possible only because of intercalary meristem. Clear? Okay. Now this was about primary meristem. Now coming into secondary meristem, as we have discussed, it is fascicular vascular cambium and Cork cambium. Okay. Now coming into permanent tissues. Okay. Permanent tissues. Permanent tissues also there are two types, mainly two types of permanent tissue. First one is simple permanent tissue. Symbol means it's symbol. there will be only one type of cells. Okay, one type of cell, so it is simple permanent tissue, and there will be complex permanent tissue where there will be more than one type of cell. Clear? Okay. So this simple permanent tissue will be having all cells similar in structure and formation. Clear? Then complex, many different types of cells will be present. Okay, simple permanent tissue, there are the first type of simple permanent tissue is parenchyma. Okay, what it is? Parenchyma. As I said, simple permanent tissue will be made up of single type of cells. Okay, so the first one in simple permanent tissue is parenchyma. For parenchyma, just remember the word TSS. Okay, what is it? For parenchyma, whenever you hear the word parenchyma, just remember the word TSS. Okay, TSS. What does the TSS indicates the function. Okay, various functions of parenchyma is indicated by TSS. What is TSS? Photosynthesis, storage and secretion. So parenchyma TSS, photosynthesis, storage and secretion. Clear? Now let's see the anatomy of parenchyma. It forms the major component within the organs. Okay, it forms the major component. Then the cells of parenchyma are generally isodiametric. Means almost the shape will be almost it will be similar. Okay, isodiametric. Then they can be spherical, oval, round, polygonal or elongated in shape. They can be any of these. Then the cells are thin. The cell walls are thin. This is a point to be noted. The cell walls are thin and it is made of cellulose. Clear? Now the cells may be either closely packed. The cells can be either closely packed or they can have intercellular spaces between them. Now the functions we have already discussed, it is photosynthesis, storage and secretion. Clear? Now coming into the second type of simple permanent tissue, it is collenchyma. What it is? It is collenchyma. Okay, so collenchyma occurs in layers below the epidermis. Okay, where this parenchyma was seen in majority of the places, but collenchyma is present in the layer which is below the epidermis. Clear? Now we will see about the features of collenchyma. The features of collenchyma will be okay, the features of collenchyma. The features of collenchyma, it will be having thickened corners. It will be having thickened corners. Then it will be having protoplasm, a vacuole. Okay, the corners will be thickened. The cell wall will be arranged, and it will be having protoplasm and vacuoles. Clear? Okay, so that was about secondary, the simple tissue, simple permanent tissue. Okay, first one was parenchyma, second one was collenchyma, and third one is sclerenchyma. What is the third one? The third one is sclerenchyma, and in sclerenchyma again there are two types. Okay, in sclerenchyma there is two types. It's scleroid. Okay, one is scleroid. The scleroid will be formed of, the scleroid will be having pitch. The scleroid will be having pitch. It will be having lumen and thick cell wall. These are the cells of sclerenchyma. Sclerenchyma are of two types and both are dead tissues. What are they? They are dead tissues. Okay, and they will be having pitch, lumen and thick cell wall. Okay, the scleroids are spherical, oval, cylindrical, and they will be having high thickened dead cells. Okay, thickened dead cells will be there and very narrow cavities which is known as lumen. These are not so important as per NEET perspective. These are for your basics. Okay, just remember that there are two types of sclerenchyma cells. Sclerenchyma. Okay, now where these are found? This was a PYQ, I think. Okay, so fruit walls of nut. Sclerenchyma is present in fruit walls of nut, pulp of fruits and seed coat of legumes. Seed coat of legumes and leaves of tea. Clear? Okay. Now coming into complex tissue. As we said, complex tissue will be possessing more than one type of cells. Okay, are made up of more than one type of cells which work together as a unit. They will be formed by more than one type of cells, but they will be working as a single unit. Okay. Now xylem and phloem will constitute complex tissue. Xylem plus phloem will constitute the complex tissue. Now xylem. First we will be discussing about the xylem. Xylem is a conducting tissue. Xylem is a conducting tissue. What do you mean by conducting? Conducting means they take the water, gases, the food here and there. So that transport system is known as conducting. So xylem is the conducting tissue for water and minerals from roots to stem and leaves. So it conducts water and minerals from roots to the stem and leaves. Clear? And it also provides mechanical strength. It also provides mechanical strength to the plant. Now what are the different kinds of elements present in xylem? There are 4 types of elements. As we said, complex tissue will be made of more than one cells. So there are 4 different kinds of elements in xylem. Now what are the 4 different types? Just remember TVFP. TVFP. Tracheids, vessels, xylem fibers and parenchyma. Xylem parenchyma. So 4 tissues, 4 elements. Okay, there are 4 elements. There are 4 elements. The first one is TFVP. Just remember TFVP. Tracheids, vessels, xylem fibers, xylem parenchyma. Clear? Okay, now discussing more about the tracheids. This is a point to be noted. This was a PYQ. Gymnosperms lack vessels in their xylem. Gymnosperms will be lacking xylem in their vessel. They are just having phloem. They don't have xylem. I mean, they are having xylem, but their xylem will be lacking vessels. They won't be having vessels. Okay. So tracheids. The main property of tracheids is that they are dead. Okay. Tracheids are elongated or tube-like cells with thick and lignified walls. Now they are dead without protoplasm. Tracheids are elongated. Just remember this picture. Whenever you hear the word tracheid, remember the picture. Okay, these are elongated structures. Now the inner layers of cell walls. Inner layers of cell wall will be having thickenings. Okay, the inner layers of cell wall. You can see, you can appreciate it. There will be thickening. Okay. Now in flowering plants, both tracheids and vessels will be found. In gymnosperm, please note the point. In gymnosperms, vessels are absent, but in flowering plants, both tracheids and vessels will be present. Okay. Now vessels. Vessels are also dead. Vessels are again dead. Vessels are long cylindrical tube-like structures. Just remember this. These are elongated tube-like structures. But these are cylindrical tube-like structures. Okay. Made up of many cells called vessel members. What are the cells present? Vessel members. And each will be having lignified walls and a central cavity. This structure, the vessel structure, is having a central cavity. As you can appreciate, there is a central cavity in this vessel. Okay. Now the vessel cells are also dead and are devoid of protoplasm. The vessel members are interconnected through perforation in their common walls, which means the vessel members, there are so many, this vessel is made up of so many vessel members, right? And these vessel members are interconnected because of their perforations. They are interconnected because of their perforations in their common walls. Okay, clear? And wherever vessel is present, it is an angiosperm. Clear? Because vessels is absent in gymnosperm. So this was about the xylem, tracheid and vessels. Okay. Now as we discussed, this is sclerenchyma. As I said, there are 2 types of sclerenchyma. One, it is fibers, and the second one, it was scleroids. Now coming into the third element of xylem, it is xylem fiber. What is it? Xylem fibers. Xylem fibers is again dead. It is again dead. So the 3 component of, there are 4 component. Out of these 4 component, the 3 component are dead. Which are they? They are the xylem vessels, tracheids and xylem fibers are dead. The only living, this was a PYQ, please note the point. The only living member of xylem element is xylem parenchyma. What is it? Xylem parenchyma. Clear? So just remember, xylem fibers are dead again, and xylem fibers have highly thickened walls, and they don't have central lumens. They don't have, as it is a fiber, it is just a fiber and it doesn't have a central lumen. Clear? And they can be septate or aseptate. Septate means they are separated. Aseptate means they are not separated. Clear? Okay. Now xylem parenchyma. Xylem parenchyma cells are living. So these are the only living cells in xylem elements, and they are thin walled. All the others were thick walled. This is thin walled, and their cell walls are made up of cellulose. Their cell walls are again made up of cellulose. Now they store food materials. This is an important point. They store food materials in the form of starch or fat and other substances like tannins. They store the food materials like starch or fat and other substances like tannins. So they are also a form of storage body. Then the radial conduction of water takes place by the ray parenchymatous cell. The radial conduction takes place by ray parenchymatous cell. Now we are talking about primary xylem. So this was all the 4 elements of xylem. Now there is another entity known as primary xylem and secondary xylem. Okay, so we will be discussing about primary xylem. Primary xylem is of 2 types, protoxylem and metaxylem. The first formed primary xylem is known as protoxylem and the later formed one is metaxylem. It is pretty simple. Okay, now endark. There are 2 types. Again the arrangement. Because of the arrangement, the xylem can be named into endark and exarch. In stems it is endark, which means the growth of xylem is towards inside. So it is endark, and which means the protoxylem will be towards the inside, towards the pith. So if the protoxylem lies towards the center, it is known as endark. Let's see. This is protoxylem. This is the first formed xylem. Okay, this is the later forming xylems. But this first formed xylem is directed towards the pith. This is the pith. Okay, so if the first formed xylem is directed towards the pith, then it is known as endark. So endark is always seen in stem. Okay, and exarch, there is another entity known as exarch. Exarch is always seen in roots. Okay, exarch is seen in roots. What is the character of exarch? Protoxylem lies towards the periphery, which means the first formed xylem will be to the periphery. This will be the pith and this will be the first formed xylem. This will be the later formed xylems. Okay, so it is away from the pith. So it is known as exarch. So metaxylem is formed towards the center. Clear? Now coming into the next second type of complex tissue, it's phloem. Phloem is also helpful for transporting the food materials from leaves to other parts of plant. The xylem was transporting the water and minerals from root to leaves and other parts of plants, but here the xylem is returning the process, which means the made food is carried to the other parts of plants. Clear? Now the first formed primary phloem will be called as usual protophloem, and the later formed phloem has bigger sieve tubes. Okay, the later formed phloem will be known as metaphloem. Now let's see about the characters. The first formed primary phloem or protophloem will be having narrow sieve tubes. What it will be having? It will be having narrow sieve tubes, and the later formed phloem will be having bigger sieve tubes. So there is one big difference. It is that the protophloem will be having narrow sieve tubes and the metaphloem will be having bigger sieve tubes. Clear? Okay. And in angiosperms, just remember this. In angiosperm, phloem will be having SCFP, which means they will be having sieve tube elements, they will be having companion cells, they will be having phloem fibers and phloem parenchyma. As we discussed in xylem, there are 4 elements. In phloem also there are 4 elements. The first one is sieve tube element, the second one is companion cells, third one is phloem fibers, and the fourth one is phloem parenchyma. Clear? And in gymnosperms, the phloem will be having SAFP, which means they will be having sieve tubes, they will be having albuminous cells, they will be having phloem fibers and phloem parenchyma again. So in common they will be having sieve tube elements, phloem fibers and phloem parenchyma. In angiosperms they will be having companion cells, whereas in gymnosperms they will be having albuminous cells instead of companion cells. And there is one more point to be noted. These sieve tube elements and companion cells are formed together. They are found together. They are dependent to each other. Clear? So this was all about the basics of anatomy of flowering plants. Now we are going into the detail of the chapter as per the new NCERT. So what is basically anatomy? It is the internal structure and functional organization. We are studying about the internal structure. Okay, so that is anatomy. So internal structure, study of internal structure of plant is called anatomy. Now tissue system. There is tissue system. What do you mean by tissue system? When cells are formed, different types of cells are united together to form, the types of cells are united together to form a tissue system. Okay, they will be performing a function, they will be performing a common function. So tissue, let us see that their structure and function would also depend on the location. So depending on the location of the structure and function, there will be different types of tissues. Okay, so there are three types of tissue system. There are three types of tissue system. Let's see the epidermal tissue system, the ground or fundamental tissue system, and vascular or conducting tissue system. So there are three types of tissue system: epidermal tissue system, ground or vascular, ground tissue system. That's enough. Then vascular or conducting tissue system. You just have to remember the basic words, and if they ask by the other name also, you have to answer it. It's epidermal, ground and vascular. Okay, first we will be dealing with the epidermal tissue system. Epidermal tissue system will be tissue system forms the outermost covering of the whole plant body. So epidermis is the outermost layer. Whenever we study about our human body itself, human body or animal, it's epidermis. Epidermis is the outermost layer. Epidermis is the outermost layer. So in plants also, epidermal tissue system is the outermost part of plant body. Clear? And it will comprise of epidermal cells. Clear? Stomata and epidermal appendages. So in this epidermal tissue system, there is epidermal cells, stomata and epidermal appendages. So let's see each of them. The epidermal appendages, there are two epidermal appendages. They are known as trichomes and hairs. What are they known as? They are known as trichomes and hairs. Now the epidermis is the outermost layer, as we said. It is made up of elongated, compactly packed. It is made up of elongated, compactly packed, arranged cells. Okay, which form in a continuous layer. So these epidermal cells are also formed by elongated cells. They are also arranged continuously. They are always single layered. These epidermal layer is always single layered. They are parenchymatous. Please note these characteristics as they are important. Single layered, parenchymatous, and there will be small amount of cytoplasm in the cell wall. Clear? And the outside of epidermis. Epidermis is the outermost layer, but in the outer part of epidermis also there will be another layer which is protecting the epidermis. So the protection of epidermis is done by another waxy layer. Waxy means waxy coating. They will be looking like a waxy coating. Okay, epidermis is often covered with a waxy thick layer which is known as cuticle. Okay, what is the function of cuticle? It will prevent the loss of water. The function of cuticle is prevention of loss of water. Okay, now the cuticle will be absent in roots. As we said, the primary plant body means the total plant body, the outer surface will be covered by epidermis, and the epidermis will be having a protective layer of cuticle, but it is not present in the root, which means root is absent of cuticle. Everywhere else cuticle is present. Clear? Now, as we said, there will be stomata. What is stomata? They are the structures present in the epidermis of leaves. In the epidermis of leaves, they are also present. They will regulate the process of transpiration and gaseous exchange. So this is a MCQ point. Stomata is responsible for the transport of transpiration and gaseous exchange. They are responsible for the transpiration and gaseous exchange. Now this structure, which is known as stomata, will be composed of two bean-shaped cells known as God cells. Okay, so the stomata will be composing of two bean-shaped structures which is known as God cells, and which will enclose the stomatal pore. These God cells will be formed together to have they will be enclosing to have a stomatal pore. We will see the picture. So just see this. There are two bean-shaped structures. These are the bean-shaped structures. This is the stomata. Okay. So this is the stomata, and there are two bean-shaped structures formed. These bean-shaped structures are enclosed to form this stomatal pore. This is the stomatal pore. Clear? And just see that. Just see the inner layer. inner wall of God cells. Inner wall of God cells are thicker when compared to the outer walls of God cells. The outer walls of God cells are thinner layer, whereas the inner walls of God cells are thicker layer. Clear? And in grass, as we said, it will be having bean shaped God cells are present usually, but in grass, which means in monocots. Grass is a monocot, right? So in grass or in other monocots, the God cells are dumbbell shaped. What it is? It is dumbbell shaped. Now the outer walls of God cells, as I said, it will be thin away from the stomatal pore, and inner walls, it will be highly thickened. Now the God cells also have chloroplast. Just see the picture clearly, you can see some specific green structures. These are the chloroplast. So the God cells is also having chloroplast. They are also possessing chloroplast. They regulate. Now the function of God cells. What is the function of God cells? They will be regulating the opening and closing of stomata. They will be regulating the opening and closing of stomata. Now the epidermal cells, these are a part of epidermal tissue, right? So the epidermal cells which are present subsidiary, which means which are present near to the stomata, can be modified to form subsidiary cells. So just remember what are subsidiary cells. They are the modifications of nearby epidermal cells. Okay, they are the modification of nearby epidermal cells near to the stomata. So in the vicinity. So a few epidermal cells in the vicinity of God cells become specialized in their shape and size to form subsidiary cells. So this stomatal aperture plus this God cells and surrounding subsidiary cells all together can be forming the stomatal apparatus. So it is the stomatal apparatus. And just see the picture, it will be having a God cell, stomatal pore, chloroplast, subsidiary cells and epidermal cells at last. So whenever you see the picture, you can get the information about the whole paragraph here. Just study the picture, you will get all of it. Okay. Now as we said, they will be having epidermal appendages. Cells of epidermis will be having number of hairs, and these root hairs, there are two types, trichomes and hairs, root hairs. Okay. So root hairs are unicellular. Just remember the point. Root hairs are unicellular. They are unicellular and they absorb water and minerals from the soil. And in stem, in root it was root hairs, and in stem the epidermal hairs are known as trichomes. What are they known as? They are known as trichomes, and they are usually multicellular. So please note the point. Root hairs are unicellular. What are they? They are unicellular, whereas the trichomes are multicellular. Clear? Now what are the function of trichomes? They can even be secretory, and they help in preventing water loss due to transpiration. They help in prevention of water loss. Clear? Now the first tissue, epidermal tissue system is over. Now coming into the second type of tissue, which is ground tissue system. What it is? It is ground tissue system. Okay, all tissues except epidermis and vascular bundles constitute the ground tissue system. So we are basically learning about three types of tissue system: vascular tissue, ground tissue and epidermal tissue. So the epidermal tissue and vascular tissue, the all the tissue which are present except epidermal and vascular constitute the ground tissue. Clear? Okay. And so except epidermis and vascular tissue, all tissues except epidermal tissue and vascular tissue constitute the ground tissue. And what are the features of ground tissue? It will be having simple tissues. As we studied, what is simple tissue? We studied. This is what I said. For your, just for your basics, we have learned the old part of NCERT. Okay. So it will be having simple tissues like parenchyma, collenchyma and sclerenchyma. Clear? And now the parenchymatous cells are usually present in cortex, pericycle, pith and medullary rays. Wherever the parenchymatous cells are present, they are present in the cortex, pericycle, pith, medullary rays. All these structure we will be studying in detail in the next page. Okay. And in the primary stems and roots. So the parenchyma is present in all of these places, Where are they? Cortex, pericycle, pith, medullary rays, and the primary stems and roots. Okay. And in leaves and ground tissue consist of thin-walled chloroplast. In leaves, the ground tissue will be having a thin-walled chloroplast, and they are known as mesophyll. Okay, they are known as mesophyll. Just remember the point. There is a structure known as mesophyll. What is mesophyll? They are the thin-walled chloroplast containing cells. They are the thin-walled chloroplast containing cells. They are chloroplast containing cells present in the ground tissue. So now coming into the vascular tissue system. So this was about ground tissue system. In ground tissue system, we just studied that it is made up of parenchyma, collenchyma and sclerenchyma. Clear? Now coming into vascular tissue system. Vascular tissue system, as we said, it will be the conducting system which conduct the water and minerals to the leaves and food and other things from the leaves to the other parts of body. Clear? So they are made up of complex tissue. Ground tissue is made up of simple tissue, whereas vascular tissue is made up of complex tissue, which are the phloem and xylem. Okay, xylem and phloem together constitute the vascular bundles. In Dicots stems there will be cambium. Cambium is present between phloem and xylem. So just remember this point. In dicots there will be presence of a new structure which is known as cambium. What is cambium? This is, just remember that, just think that this is xylem and this is the phloem. So between the xylem and phloem there is a structure. If there is a structure between the xylem and phloem, that is known as cambium. Okay, that is known as cambium, and this cambium is responsible for the secondary formation. Okay, secondary tissues formation, and this is only found in dicots. This property is only found in dicots. So cambium is responsible for the secondary tissue formation, and cambium is only present in the dicot plants. Clear? So in dicot stems, cambium is present between phloem and xylem, and such if this cambium is present, they are able to grow, right? They are able to do their secondary growth. So they are known as open vascular bundles. They are known as open vascular bundles. Vascular bundles, because of the presence of cambium, they have the ability to form secondary phloem and secondary xylem. So they are known as open vascular bundles. Whereas in monocots, this cambium is absent. They don't have, the vascular bundles have no cambium, so they do not form the secondary tissues. Okay, and they are referred as closed vascular bundles. So this was about that. Now xylem and phloem. There are two types of arrangement of xylem and phloem. See here, the xylem and phloem are arranged in different radii. Means this is one radii and this is another radii of the circle. So they are arranged in different radii. If the xylem and phloem are arranged in different radii, then they are known as radial arrangement. What are they known as? Radial arrangement of xylem and phloem. Just see, if they are arranged in alternate manner along different radii. Okay, different radii. This is the point to be noted. Different radii. They are known as radial arrangement, and these type of arrangement are seen in roots. This type of arrangement are seen in the roots. Always these type of arrangement are seen in roots. And if they are conjoined, what do you mean by conjoined? This is conjoined, means they are on the same radii but one after another. So they are known as conjoined. They are joined together. The xylem and phloem jointly situated along the same vascular bundle, along the same radius of vascular bundle, then they are known as conjoined arrangement, and this is seen in stems and leaves. So in roots it's always radial type of arrangement, and in stems and leaves it's always conjoined type of arrangement. And in conjoined the phloem will be present outer of the xylem, which means the xylem will be inside and the phloem will be outside. Okay, phloem, just remember by this technique, xylem, phloem, right? This phloem is having O, which means phloem is outside and xylem is inside. Anatomy of, now we have studied about ground tissue vascular tissue and epidermal tissue. Now connecting all these information we have gathered, we are going to study about dicot stem, monocot stem, dicot root, monocot root, and the dicot and monocot leaves. So let's go into it. First one we are dealing is dicot root. Okay, dicot root, for example, we will be taking sunflower. Okay, transverse section of sunflower root. So the outermost layer, it is epidermis, right? So here it is known as epiblema. What it is known as? It is known as epiblema. So the outermost layer is epiblema. Now many cells of epiblema will be protrude in the form of unicellular root hairs. As we have discussed, this epidermis will be having epidermal appendages and they are known as root hairs. If they are present in the root, they are known as root hairs. And so the epidermal tissue is over. This is the epidermal tissue and that got over. Epidermal tissue got over. Now comes the second one, which is ground tissue. Ground tissue starts with the cortex. Okay. So the cortex will be consisting of several layers of thin-walled parenchyma cells. As we have discussed, the ground tissue is formed of simple cells, parenchyma, collenchyma, sclerenchyma. So here in dicot root, it will be having several layers of thin-walled parenchyma cells. Several layers of thin-walled parenchyma cells with intercellular spaces. And the innermost layer of cortex is called endodermis. What is the innermost layer called? The innermost layer is called endodermis. And it will be having a single layer of barrel-shaped cells. These inner to endodermis, they will be having a single layer of barrel-shaped cells. Okay, barrel-shaped cells they will be elongated. Okay, without any intercellular space. Then the tangential as well as radial walls of the endodermal cells will be having deposition of a waxy coating, which is known as suberin, and this is known as Casparian strip. This is known as Casparian strip. So this we will be discussing. Just listen to this part. So this is the epiblema. This is the epidermal layer. The first layer that you see is the epidermal layer. Then outer to the epidermal layer you will be seeing root hairs. What did you see? Root hairs, which are the epidermal appendages. Now inner to the epidermis you have seen cortex. Cortex will be having arranged parenchymatous cells, linearly arranged parenchymatous cells with little intercellular spaces. And in the innermost layer of, as they said, the innermost layer of cortex. Innermost layer of cortex is known as endodermis. What is they known as? So this layer, this pink colored structure, the innermost layer of cortex is known as endodermis. What are they known as? They are known as endodermis. And in this endodermis, in their walls, this is endodermal cells. These are endodermal cells. Okay, these are endodermal cells, and these endodermal cells are arranged without any intercellular space. And this endodermal cells will be having thickened walls. They will be having thickened walls due to a material which is known as suberin. They will be having thickened walls. They will be having thickened walls because of a material known as suberin. What is suberin? Suberin is a water impermeable substance, which means the material won't allow the water to pass through it. So suberin is a water impermeable material, and this suberin is seen along the walls of endodermal cells. So suberin is present, and this type of arrangement, it will look like a strip. So it is known as Casparian strip. What it is known as? It is known as Casparian strip. So Casparian strip is formed of, this is an MCQ point, Casparian strip is formed of suberin. So next to endodermis. So this before endodermis there was thin-walled parenchyma. So inside the endodermis there is thick-walled parenchyma. Next to endodermis lies a few layers of thick-walled parenchymatous cells, and these thick-walled parenchymatous cells are referred to as pericycle. What are they referred to as? They are referred to as pericycle. So pericycle is having thick-walled parenchymatous cells. And initiation of lateral roots and vascular cambium is taking place in these cells. So this was a PYQ. This was itself a PYQ before. The initiation, what is the origin of lateral roots? The origin of lateral roots are from pericycle. Clear? Initiation of lateral roots and the vascular cambium is from pericycle. And the secondary growth also, since cambium is there, secondary growth also takes place from there. The vascular cambium origin is pericycle. Just remember that point. Now the pith. Pith is the inside. The sender. The sender is pith. This is the pith. This is the pith. As you can appreciate, this is the pith. This is the pith. Okay. So here the pith is inconspicuous. Means it's not so significant. Here the pith is in dicot root. We are discussing about dicot root. So in dicot root, the pith is not significant. Now the parenchyma cells. The parenchymatous cells which are present, between the xylem and phloem are conjunctive tissue. So what are conjunctive tissue? The parenchyma cells which are present between the xylem and phloem. Between the xylem and phloem. Okay, they are known as conjunctive tissue. They are not the cambium. Cambium was between the xylem and phloem. Between the arrangement of xylem phloem. Here some parenchymatous tissue are arranged between xylem and phloem. They are known as conjunctive tissue. Okay. Now there are usually 2 to 4 xylem and phloem patches. Means 2 to 4 pairs of xylem and phloem. 2 to 4 pairs of xylem and phloem are present in dicot root. And cambium ring will be developed between the xylem and phloem. Later there will be formation of cambium ring between xylem and phloem. And all tissues on the inner side of endodermis. All tissue, as we have discussed, till here it was cortex. Till here it was cortex, and the innermost layer of cortex was endodermis. So all the tissues present inside the endodermis, including the pericycle, vascular bundles and pith will constitute the stele. So what is stele? Stele is equal to pericycle plus vascular bundles plus pith. So pericycle plus vascular bundles plus pith plus endodermis. All the endodermis won't be there. All the cells, all the tissues present inner side of endodermis, like pith, pericycle and vascular bundles, are known as stele. Are together known as stele. Clear? So this was about dicot root. Now coming into monocot root. Monocot root, the anatomy of monocot root is similar to the dicot root in many aspects. It's almost the same. The only difference we will be discussing here. It has an epidermis, it has a cortex, it has an endodermis as the same. Cortex, epidermis, cortex, endodermis, pericycle, vascular bundles and pith. Everything is the same. The only difference is that they are usually having more than 6 xylem bundles. There it was 2 to 4. In dicot root it was 2 to 4 pairs of xylem phloem patches. But here it is always more than 6. It is always more than 6 xylem bundles. Clear? And pith is large. There the pith was not significant, it was small. Here the pith is large and well developed. Clear? And the monocot do not undergo any secondary growth. In dicots there is always presence of cambium and there will be secondary growth. In monocots there is no cambium and no secondary growth. Now going into dicot stem. Before that we will see the picture. It is again monocot root. The cortex, endodermis, phloem and xylem. There are more pairs of phloem and xylem bundles and the pith is large. Clear? Going into dicot stem. Dicot stem, as we know, the first thing, the outermost layer will be epidermis. And the outermost protective layer of the stem is epidermis. And they will be covered by a thin layer of cuticle. We studied this. And they may bear trichomes. What are trichomes? They are the appendages present in the stem. Epidermal appendages or epidermal hairs present in the stem. Okay. And they also have few stomata. Means they will also be having some stomata. Then the cells are arranged in multiple layers between the epidermis and pericycle. Okay. So the cells arranged between epidermis and the pericycle are known as cortex. So what is cortex? The cells arranged between the epidermis and pericycle are known as cortex. It consists of 3 subzones. There will be 3 subzones. Here the cortex is divided into 3. Here the cortex are divided into 3. Let's see that. So first we will discuss the theory and see the picture so that it will be clear. So the 3 subzones are outer hypodermis, then collenchymatous cells, So the first layer is outer hypodermis, then it will be cortical layers, and the third one will be endodermis. So the three subzones are outer hypodermis, then middle cortical layers and inner endodermis. Clear? Now what is hypodermis? This hypodermis will be consisting of a few layers of cholangimatous cells. Few layers of cholangimatous cells are present in the outer hypodermis. And this, what is the function? Mechanical strength to the young stem. So the cholangyma are having corner thickened cell wall, right? Corner thickened cell wall. Thickened cell wall are the feature of cholangyma. So the cholangyma will be providing mechanical strength to the body of plant. Now cortical layers. Cortical layers below the hypodermis. Below the hypodermis there is next layer, next zone, which is cortical layers, and it will be consisting of parenchymatous cells. Okay, they are having parenchyma. So hypodermis is having cholangyma. Cortical layer is having parenchyma. And now the innermost layer is known as endodermis. Endodermis is rich in starch grains. Okay, they are rich in starch grain, so they are also referred to as starch sheath. They are also referred to as starch sheath. So up to this we will just rewind. Till here we will rewind. First the epidermis, then the cuticle, the protective layer, then they will be having trichomes and stomata. They can be having trichomes and stomata. Then the cells arranged between the epidermis. This is the epidermis, and between the epidermis and pericycle consists the cortex. So this cortex is divided. Okay. This is the cortex, and the cortex is divided into three subzones. The first one is the outer hypodermis, and hypodermis is made of cholangyma. The second one was the cortical layer. What is the second one? It is cortical layer. It is made of parenchyma. And the third one is endodermis. Endodermis is made of parenchyma, but they also are rich in starch. So they are also referred to as starch sheath, and this was an MCQ, previous year question. Okay, so they are also known as starch sheath. Starch sheath is present in, or starch sheath is a characteristic of dicot stem. Clear? And now comes the pericycle. Okay, before going into pericycle, we will just see the picture. This is epidermis, cholangyma, parenchyma of cortical layer, cholangyma of hypodermis, then endodermis. Okay, now coming into pericycle. Pericycle present on the inner side of endodermis, and it will be present above the phloem. Okay, and they will be present in a semi-lunar patch of sclerenchyma. So the pericycle is made up of, here the pericycle in dicot stem, the pericycle is made up of sclerenchyma. Okay, the pericycle is made up of sclerenchyma, and in between the vascular bundles there are few layers radially placed parenchymatous cells. As we seen in the dicot root, conjunctive tissue between the xylem and phloem, which is parenchymatous. Here also there are parenchymatous cells between the xylem and phloem, and here they are known as medullary rays. What are they known as in dicot stem? They are known as medullary rays. So medullary rays, just remember these specific words: starch sheath, medullary rays, conjunctive tissue. All these are particular to particular things. So medullary rays are always in dicot stem. And a large number of vascular bundles. Are arranged in a ring. So I think this was the PYQ. Numerous rings like arranged vascular bundles are seen in dicot stem. Clear? Okay. And the bundle is conjoined, which means xylem inner and phloem outer. Okay, in the same radii, and they will be open because of the presence of cambium and the endark protoxylem. What do you mean by endark? Endark means the protoxylem is directed towards the pith. And there is a large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion and constitute the pith. So the pith is large and significant here, and the pith is also made up of parenchymatous cells. Now see the picture clearly. This is the pericycle. What is the pericycle made of? Sclerenchyma cells. And inside the pericycle, what is the location of pericycle? Above the pericycle it is endodermis, and below the pericycle it is phloem, or the pericycle is found above the phloem. Okay, so this is phloem, and as we said, the xylem and phloem are conjoined. Xylem and phloem are conjoined, which means they are in the same radii. Okay, same radii, phloem and xylem, and between the phloem and xylem there will be cambium, so there is secondary growth. And the protoxylem is directed towards the pith. So it is endark. Clear? So this was about dicot stem. Now coming into, and the pith is also large. Now coming into monocot stem. And just remember this thing, this is ring-like structures. Ring-like structures are also present in dicot stem. And the space between, this is the cells present between the xylem and phloem. Okay, this cells which is present between the xylem and phloem, between the vascular bundles, they are known as medullary rays. Okay, medullary rays. And please note the picture again. Now coming into monocot stem. Monocot stems are, as we studied, there are 3 subzones in stems. Here the hypoderma. In dicot stem, the hypoderma was made up of collenchyma. But here in monocot stem, the hypodermis is made up of sclerenchyma. Sclerenchymatous hypodermis and scattered vascular bundles. There it was arranged ring-like arrangement of vascular bundle. Here it is scattered vascular bundle arrangement. And each vascular bundle is surrounded by sclerenchymatous bundle sheath. So you got a point again. Bundle sheath. Starch sheath was present in dicot stem, but bundle sheath is present in monocot stem. Okay, and large pith, large parenchymatous ground tissue. Large parenchymatous ground tissue is also present, and the vascular bundles are again conjoined. Okay, in the same radii, and closed. Closed means cambium is absent, no secondary growth. And peripheral vascular bundles are generally smaller than the centrally located ones, which means these are the vascular bundles which are scattered in arrangement. Okay, the vascular bundles are scattered, are arranged scattered. Okay, so this is the hypodermis which is made up of sclerenchyma, and this is the vascular bundles. These are the vascular bundles, and vascular bundles which are placed periphery will be larger, and the one placed in center will be small. Right? Peripheral vascular bundles are smaller. And the central vascular bundles will be larger.